Hello again. This is take 2 of lecture 11D. We went through the first time. I didn't have my mic down, and so the audio was really, really faint. So let's try this again. Okay, we're kind of taking a little bit of a detour from what we've been talking about so far. We just got through talking about meiosis. And there's another set of phenomena that you might be aware of, in which you might have to do some explaining at some point, being a biologist. These are issues that are of some concern to many people, and this is really the best place for us to fit it into our discussion. And so let me see if I can put a little bit of context in this. A few lectures ago, we talked about the cell cycle and the cell cycle's checkpoints. And there was a G1 checkpoint, G2 checkpoint, then there was the M checkpoint. And I said that the M checkpoint was really important because it's the place where you are basically making sure that the chromosome, this is our chromosome that is there at the metaphase plane. It's a doubled chromosome because it's got two sister chromatids. We need to have the spindle fibers attaching to the centromeres correctly, so that when the spindle fibers start to result in the chromatids moving apart from each other, this chromatid will be moving in that direction, this chromatid will be moving in that direction, the right way, right? And if you do that, then we end up with exactly the same number of chromosomes in both daughter cells, right? Now, what happens, what we're concerned about here is, well, what would happen if we have spindle fibers attaching on one side? Here's that same chromosome. If we have the spindle fibers attaching on one side, but we haven't quite attached spindle fibers over here, right? Well, we missed. The spindle fibers never make the connection between centriole and the centromere here. What's going to happen is that both of these chromatids are going to go off in the same direction, right? In other words, you're going to have, two chromosomes, two chromatids going this way. Each one will become its own chromosome, right? And on the other side, you'll have 0. And if this were happening in your cells, remember your cells normally have 46 chromosomes total. If that's a normal number, what would happen is that the cell, the daughter cell rising over here, would have 47, right? And the cell over on the right-hand side would have 45, right? And so what we're saying here is that if we don't get the correct segregation of the chromosomes during mitosis, during anaphase, right? That's when the chromosomes split apart, right? The chromatids split apart, right? Then we end up with the incorrect number of chromosomes, right? And that, I'm hoping, makes pretty good sense to you, right? Because I wanted to say things in those words first, before I introduced a couple of big words, things that we have, words that get tossed around a lot in like chapter 8 and our subsequent discussions in BIO110. And these two words are non-disjunction and aneuploidy. Right? But you know what these mean, right? Both of these terms are completely clear to you right now, because you understand that if you don't get the spindle fibers attaching correctly to the chromatids during metaphase, right? In other words, if we make a mistake at the metaphase checkpoint, what's going to result is an incorrect chromosome segregation. And so incorrect chromosome segregation okay, I know it's not terribly legible, but you can read it right here. Incorrect chromosome segregation is what we mean by non-disjunction. Right? Kind of makes sense. I mean, you can say, well, disjunction sounds like we're separating them apart, right? Non-disjunction means that we're not separating them. In other words, both chromatids are going in the same direction, right? It's a good way of thinking about it. And if you have non-disjunction, if you have the incorrect chromosome segregation, what you end up with is not the right number of chromosomes, right? And so, well, A before something means not. U means correct or true, right? Ploidy refers to the number of chromosomes. So aneuploidy literally means not the right number of chromosomes. Okay. So that's basically hopefully useful to some of those of you who struggle with the terminology. I know that's not everybody, but that's something that people will tend to struggle with more until you start realizing that knowing a little bit of Greek and Latin is going to go a long way when it comes to understanding some of these elaborate words in biology. Okay. All right. Now, why are we talking about this now as opposed to when we were actually discussing mitosis? Well, it's a good reason, okay? Because non-disjunction, when it occurs during mitosis, I mean, it does occur. I mean, M checkpoints are really good, but clear mistakes get made. And if a mistake gets made at the M checkpoint of mitosis, what you're going to end up with is a daughter cell that has the wrong number of chromosomes on both sides of the metaphase plane. In other words, we'll end up with aneuploid cells. But during mitosis, you have two aneuploids, two cells with the wrong number of chromosomes. Out of several trillion, you will have trillions of cells in your body. And if two of those cells end up with the wrong number of chromosomes, it's no big deal, right? You've got plenty of cells, two cells, no big deal. Okay. The other thing is that if non-disjunction occurs in your fingertip and you end up with these two aneuploid cells in your fingertip, they might actually stay with you for the rest of your life, right? But one thing that's not going to happen is that they're not going to be passed on to your offspring, because the only cells that you pass on to your offspring, the only nuclei that you pass on to your offspring are the nuclei that are there in the gametes. And so if a mutation occurs of any type, non-disjunction or otherwise, if it occurs in any cell besides your gamete, you don't have to worry about that affecting your offspring because it's going to die with you, right? Okay, so that's kind of like why it's not such a huge important thing in the context of mitosis, right? Now, if non-disjunction occurs in a meiotic cell division, right, during meiosis, the cells that result are gametes, right? And so if you end up with a gamete that's got the wrong number of chromosomes, that's going to be kind of big, right? Because if a sperm cell carries 24 chromosomes instead of 23, and it fuses with a normal egg that's got 23 chromosomes, the zygote is going to start off life with 47 chromosomes, and all the cells that come from that zygote are going to have that same number of chromosomes. In other words, you affect the entire baby, the entire adult that develops from an aneuploid zygote, right? And so whereas two cells out of several trillion is going to give you completely negligible results, right? The results, the consequences for a non-disjunction event occurring during meiosis is going to be far more severe, right? And the aneuploid conditions that are named occur as a result of usually some type of non-disjunction event occurring during the meiosis. Okay? If it's not actually passed on from the parents. If a parent is aneuploid, then the aneuploidy could be inherited by the baby. Right. So that's the context here. This is why aneuploidy, that's why non-disjunction in meiosis is of great consequence, and that's why we're talking about it on the heels of our discussion of meiosis. All right. Now, while we're on this topic, we should address what we mean by the normal human karyotype. Okay, the karyotype refers to the array of chromosomes that you find inside of a normal diploid cell. Right, and so the normal human karyotype consists of 22 pairs of autosomes. These are the chromosomes that are not related to sex determination. 22 pairs of autosomes plus the sex chromosomes, which might either be an X and an X or an X and a Y. Okay? And so if you have 22 pairs of autosomes, it's 44. 2 sex chromosomes, that's how you end up with a 46 total, 2n equal to 46. Right? Now, when we look at the number of chromosomes and we see something that is aneuploid, so for in other words, if you have an extra chromosome 21, right? This would, one, lead to a karyotype in which we have 47, not 46 chromosomes, right? It's also going to lead to a condition, a name condition in this case, that would be Down syndrome, right? And so the karyotype is one way of expressing an alteration to the normal genome occurring as a result of non-disjunction or a normal or a state of aneuploidy. Okay, so now would be a good time for me to shift over to our textbook. And the first figure is basically a nice, pretty figure of non-disjunction occurring during meiosis. It could occur in meiosis 1, it could occur in meiosis 2. The point is that the normal number of chromosomes in a gamete, okay, is something that we get, we could get. Okay? If we had completely normal meiosis, all the gene products, all the gametes would have, in this case, 2 chromosomes, a little one and a bigger one. Right? Okay. In the case of non-disjunction, we're always going to be ending up with gametes that have either the wrong number of chromosomes with 1 extra or the wrong number of chromosomes with 1 too few. Right? And so the presence of these abnormal gametes is what leads to the abnormal number of chromosomes in the zygote, which leads to conditions like Down syndrome, Klinefelter syndrome, okay, et cetera. Okay. So all of those aneuploid conditions are identifiable because of the wrong number of chromosomes. How do you detect them? Well, glad you asked, because I've got an answer for that. You can actually detect the incorrect number of chromosomes pretty easily. I mean, it's not easy, smeezy. You couldn't do this without some sophisticated equipment. But if you were to get a sample of baby's blood, apply the appropriate preservatives like colchicine to make things get arrested in mitosis, and then you were to get a nice, good, squished out copy of a picture of all the chromosomes. Okay? You would actually be able to have a computer arrange those chromosomes out so you can see them and determine whether we've got the correct number of chromosomes, like it's shown here, or whether you've got the incorrect number of chromosomes. Go back to my drawing functionality. And so looking at this, we've got chromosome 1, chromosome 2, all the way down to chromosome 22. Right? And you're probably noticing that the numbering of the chromosomes is in order of size. Right? Chromosome 1 being the largest, chromosome 22, autosome chromosome number 22 being the smallest. Okay? Sex chromosomes don't count, right? The Y chromosome is really, really tiny, but the X chromosome has got quite a few genes. It looks like this X chromosome here looks like it's about the same size as chromosome 8, right? So chromosome 8 is a pretty big, large chromosome, and the X chromosome is also a pretty big chromosome. So there's that. But because it's a sex chromosome, it's not included in the numbering system. Right? Okay. So we go through this process. One way of detecting for absolute certain that a baby is or is not trisomic or aneuploid is to get the karyotype of the baby. And if there were an extra copy of chromosome 21, that would tell you that you've got a Down's baby. If you don't have that, there's no guarantee that you're not going to have a Down's baby. There are other ways that's going to happen. At least you've got some assurance that you don't have an aneuploid. Okay. So yeah, this karyotype, number of chromosomes, that's I think the only other picture that was worth sharing from the textbook. Go back to my whiteboard now. Okay. And the thing I think what I need to get to now is maybe address to a certain degree the question of why is aneuploidy bad, right? Why does it have negative consequences on the health of the aneuploid individual? You know, why do we have the deficiencies, right? And Down syndrome, and it's actually even more severe for other types of aneuploidy. I mean, basically, if you're aneuploid in any chromosome other than chromosome 21 or the sex chromosomes, you can be aneuploid with X or Y. You can have extra X's or extra Y's. They don't have terribly severe consequences. You end up living. You can have an extra copy of chromosome 21. You have Down syndrome, but you're alive. If you do an extra copy of pretty much any other autosome, you're going to be dead before you're born, right? So in other words, aneuploidy has really severe consequences, and the best way for me to explain that is that each chromosome has its chromosomal products, and you need to have the products of the chromosomes there in the appropriate proportions. They have to have a balance in the chromosomal products needed for normal development. So in other words, if you have an extra copy of chromosome 15, right, you'll have way too much product from chromosome 2015, and then that disrupts the overall dynamic of human development, and you end up developing inappropriately, incorrectly, to the point where you would die before birth, right? And so, you know, what I'm about to say next requires you to understand that it's a bad thing to have one extra copy of a chromosome, but it's not nearly as bad to have a whole set of chromosomes extra from what you normally have, right? Because if you get a whole set of chromosomes, you're maintaining that balance of chromosomal products, right? Because you've got that balance, development tends to proceed more or less normally, and you can actually be born, you can end up having a pretty healthy life, right? And so what I'm telling you here is that if instead of having one extra chromosome, instead of actually having +1, if you were to have +23, where you had a complete copy, a complete set of all 23 chromosomes, that's normally there in a gamete, right? You might think, well, one extra chromosome is bad. 23 X chromosomes would be horrible, but no, actually, if you get a full set of chromosomes, you maintain that balance of chromosomal products, and you'll be perfectly viable. You'd be a triploid, right? Tri is the Greek prefix for 3, like triple. The 3 number of chromosomes, 3, and if you're triploid, I can't guarantee this because no human, as far as I know, has ever been born triploid. But I know with other animals, with other kinds of animals where triploidy has occurred, they tend to be perfectly viable, but they're sterile, right? They're not able to produce gametes. That makes sense because if you've got an odd number of chromosomes, you cannot undergo meiosis. Right? And so they're sterile because meiosis, which is the reduction in the number of chromosomes going from 46 to 23. If you're triploid, you would have 69 chromosomes, right? And you can't split that evenly. You're going to end up with gametes that have got totally the wrong number of chromosomes, and so meiosis is going to fail. And so, yeah, if you are triploid, you might be perfectly healthy and viable, but when it comes time for you to make babies, you're never going to be able to make any viable sperm cells or egg cells, right? Okay. So yeah, this is a phenomenon called polyploidy. Where it's kind of the wrong number of chromosomes, but instead of having one extra or two extra or one too few or two too few of the normal number of chromosomes, you get an entire extra set or an entire extra two sets. Okay, and I want to point this out because the book makes kind of a big deal out of it. If you're somehow born with two whole extra sets of chromosomes, right? In other words, if you have to double the normal complement of chromosomes, 4n tetraploid is equal to 92. Right? You would still have that balance in chromosomal products, and so there would be a reasonably good chance that you'd be viable, just like you were viable as a triploid. Right? But in the case of a tetraploid, you would also be fertile, right? And fertile because we've got an even number of chromosomes. You could actually split 4n with meiosis, and you would end up with 2n, two complete sets of chromosomes relative to what was normally there in a gamete in the original species. Does that make sense? Okay, so if you were a tetraploid and you were to mate with another tetraploid, so this is your soulmate. You found another tetraploid, you marry, you have kids, they have meiosis, giving rise to 2n gametes. And so your 2n gametes mix with fertilize their 2n gametes, and you would end up with more 4n babies, okay, just like their mom and dad, right? So yeah, this is what I mean by having fertile offspring. You could actually have babies provided you find another mate that's 4n. Okay? But if you were to mate, if you were a 4n and you were to mate with a 2n, because 2n is basically what's there more abundantly, right? As a 4n, you'd be kind of a weirdo. But if you were to mate with the original number of chromosomes, your 4n genome would give rise to a 2n gamete. Your mate's 2n genome would give rise to a haploid gamete, which means that the baby is going to be 3n. In other words, your baby is going to be sterile. Okay? Now, when you take BIO110 from me or one of my colleagues, you're going to learn that one of the things that defines a species is that you're a member of the same species if you could interbreed with them to produce viable and fertile offspring, right? So in other words, you are the same species as the person sitting across from you, because if you were somehow to interbreed, maybe not directly, but your genomes were to intermingle because of interbreeding, you'd be able to produce fertile offspring. Okay? And that's true for all humans, right? We're all part of the same species, right? But if you were a tetraploid human, right, you would not be the same species as most other humans because the offspring that you produce would be 3n and therefore sterile, right? So technically speaking, if you were to have a whole genome duplication, in other words, if you were to undergo polyploidy to the point where you have a double number of chromosomes, like that frog that's there in the example given in the textbook, that frog is a different biological species than the frogs that gave rise to it, even though there's very little difference in the genes that they contain. It's just that the tetraploid frog has twice the number of chromosomes, right? Now, this is really important for me because remember, I'm an evolutionary biologist, and I want to say very clearly that this is not the way that new species originate usually, right? On our planet, we've got millions and millions of different species. Millions and millions of different species we have today are different from the millions and millions of species we had 20 million years ago. Right? You jump to your time machine, set the clock back 20 million years, you would emerge to a different world with different species compared to what we have today. Okay, you go back 40 million years, you would see different species altogether. In other words, the history of life on our planet has been species going extinct, new species appearing to take their place, maybe increasing the number of species, maybe not. But new species origination happens all the time in the bigger picture of things. You take that long perspective. And this mechanism of going from diploid to tetraploid has happened probably a few dozen times, right? And so I do want to point out very clearly that I don't want anybody getting the wrong impression that going from diploid to tetraploid is a really important way of understanding how new species form. All right. In the next lecture, we will be moving into genetics, and I'll see you then.